Hello students, welcome. Welcome to our class. I hope you did a wonderful CET paper, right? But there were some questions which are out of syllabus. That's okay, it'll be resolved, right? We welcome you to our class, Diksha Karnataka. Now, what we're going to do is, in this class, we're going to look at the detailed explanation for the KCET biology paper, version D4, right? We have already discussed the answer keys to this paper, version D4 KCET biology paper, right? Answer keys has been discussed. Now what we are going to do in this class is we are going to look at the detailed explanation for all the questions of the biology paper, all 60 questions of the version D4, right? I'm specifically telling you it's version D4. Now, if you want the paper, right, the PDF of the paper, you can join our WhatsApp channel. Link is available in the description, right? Please join our WhatsApp channel to get the paper, right? So let's start with the question paper discussion, right? Okay, let's look at the first question. Question number 1, identify the incorrect match, right? They're asking you the incorrect match with respect to recently extinct animals and the place of extinction according to the IUCN Red List, right? Now you need to identify the extinct animal and from which place it got extinct, right? So both have to be right. Now we know the first option, doo-doo from Mauritius, this is correct, right? They're asking you the incorrect one. Please note an incorrect match. Quagga from Africa, this is also correct. Thylacine from Australia, this is correct. Now what is incorrect is stellar sea cow. Now this got extinct from Russia, right? But here what have they given? They have said it's North America, right? Therefore for question number 1, option D, right, is the right answer, right? It is the right answer. What have they asked? Incorrect match. Done. Now let us look at question number 2. Now according to the hypothesis proposed by environmental biologists, a relatively constant, right, environment in tropics promotes. Now this is discussed in, right, biodiversity and conservation, right, or organisms and population, right? Now here what are they talking about? They're talking about the latitudinal, right, latitudinal gradient. And this is under latitudinal gradient. Now what do you know about this? If you look at the earth, right, what do you see? This is equator, right? Equator. Now what is the speciality of equator? In equator what happens? There is constant environment, right? There is constant environment because the sun's rays will fall directly on the equator, right, throughout the year. Now as you go from equator towards the pole like this, what happens? You see variation. There is a lot of variation, right? Now looking at this, the environmentalists, they proposed an hypothesis. Now what did this hypothesis say? According to the hypothesis, right, speciation, right, formation of new species, it's a function of time. It is function of time. You give more constant environment and you give more time, right? Therefore here in the equator, what are species getting? They're getting more time, right? They get evolutionary time. They get evolutionary time, and millions of years, constant temperature, constant environment. Now what happens? Now species, right, species are formed, new species are formed, right? Now these species, what will they do? Right, they specialize because it's a constant environment, there is no change happening. Therefore what do? They do niche specialization, right? They do niche specialization. Niche meaning it is the address, right, the food you eat, right, the place where you live, everything. Now of the species, now what do? They specialize, right? Then this will also result in what? Greater species diversity, right? This will also result in greater species diversity. They also talk about the third point, that is here because of the constant presence of the solar energy, right, productivity is high, right? Productivity is high. These are the 3 postulates proposed by environmentalists. Now what are the asking? According to the hypothesis proposed by environmental biologists, A relatively constant environment in tropics promotes what? It will promote niche specialization and greater diversity, right? Niche specialization and greater species diversity. Option B. Option B is the right answer. Clear? Now look at question number 3. In the prevention of air pollution, right, the role of scrubber is to remove, right? The role of scrubber is to remove. Now this is a question from the deleted portion, right? The pollutions chapter is, right, the topic has been deleted, therefore this is a question from there. Now scrubbers, right, scrubbers are instruments which remove gaseous particles, right? Gaseous SO2. Now what happens? These are sprayed or passed through liquid, right, or lime water, right, or just water, right, lime water, right? Therefore what happens? These gaseous SO2 molecules are trapped, right? Therefore it is answer is gaseous SO2, right? Now this is also from a deleted topic, right? This is also from a deleted topic. Now they have given you pollutants on one side and their costs, right? What do they cost, right? Very simple question. Now you know nitrogen fertilizers, right? This where do you study? You study in eutrophication, right? Algal blooms. What is happening there? Eutrophication, nitrogen fertilizers, when they're sent, right, when they're sent into the water bodies, now what happens is it results in eutrophication. Therefore one is Q, one is Q, right? It causes eutrophication. Carbon dioxide, as you know, causes greenhouse effect, right? Carbon dioxide can trap, right, heat, therefore it will cause greenhouse effect. Now carbon monoxide is an air pollutant, right? Carbon monoxide is an air pollutant from the exhaust of vehicles also, right? Now 4 is CFCs, chlorofluorocarbons, right? They release what? They release free radical chlorines, right? Chlorines, they release free radicals, therefore they cause ozone depletion, right? They cause ozone depletion, which is P. Now do we have the answer? Q, R, S and P. Option B, right? Option B. Now look at question 5. A strict protection of biodiversity hotspots could reduce the ongoing mass extinction almost, right? To what? It is a direct question. It's 30%, right? 30%. There is no need of explanation for this question. It's directly picked from NCERT. Now another question which is memory based, right? The annual net primary productivity of the biosphere is approximately how much, right? The answer is A, 170 billion tons, right? This is also directly picked from the NCERT, right? Now look at question number 7. Now this is again from a deleted portion, right? Where you study the cycles, right? Phosphorus cycle, sulfur cycle, carbon dioxide cycle, water cycle, all of this, right? Now this is being deleted from, this is from the deleted portion. Now what do? The natural reservoir of phosphorus is rocks, right? Rocks are the natural reservoirs of phosphorus. Now this is also from a deleted portion called the succession, right? Now the sequence of communities of primary succession in water, right? If you look, this is a question they have picked from the diagram, right? There is a diagram in NCERT where you can see A, B, C, D, right? Where you can see planktons, phytoplanktons, these are floating, right? Phytoplanktons, right? After that, what do you see? You see rooted hydrophytes, right? Then you see rooted hydrophytes. From the rooted comes the free floating hydrophytes. Now these reed swamps is nothing but, right, roots are in water, right? Roots are in water and shoot is aerial. Shoot is aerial, right? Now marshy meadows is basically entire water region. Water is decreasing now and the water is completely filled with grasses, right? Grasses or vegetation, completely. Water is completely filled with water, grasses. Now then you see scrubs. Scrubs are nothing but water is decreasing now, land has started to accumulate. Now you see small trees, right? You see small trees or shrubs. Then you finally get trees, right? Or forest, right? Therefore this option D is the right answer. Okay. Now let us look at question number 9, right? Now this is also from a deleted portion called the adaptations, right? Adaptations, organisms and population adaptation. This is also from a deleted portion. Now what have they given? They have given types of adaptations on one side, then they have listed the examples on the other side. They have listed the examples on the other side, okay? Now here what do you see is, biochemical adaptation shown by deep sea fishes. Why? Because these deep sea fishes, they cannot show the behavioral adaptation. Now what is behavioral adaptation? Now if there is any problem in your neighborhood, what do? You shift to the other neighborhood. For example, right, from marine to you shift to the freshwater. Right, this is behavioral. Right, you just escape for the time being, right? This is behavioral adaptation. Now what is physiological adaptation? Physiological is you do some changes in the body, right? Some changes in the functioning of the body. That is physiological adaptations, right? What is morphological adaptations? You change the structure of certain parts of the body. You change the structure of certain parts of the body to escape the harmful or unfavorable condition. Biochemical adaptation is you do some changes in the body, right? Changes in the body permanent changes in the body, right? Producing different types of enzymes, right? Now here you can see deep sea fishes, right? They are very deep, right? So therefore they cannot do the behavioral change. Therefore what do? They do some biochemical changes by synthesizing different types of enzymes, right? They change their biochemistry, right? Therefore deep sea fishes goes for 1, right? Therefore 1 is Q. Now behavioral changes, what do you see? You see it in desert lizards. What do? If there is some unfavorable condition, they shift their place of living to another place, right? This is behavioral. Now physiological adaptations, where do you see? You see it in kangaroo rats. Now these are found in the desert regions. Kangaroo rats are found in the desert regions. Now what happens is they do not get water for a long period of time. Therefore what do? They start metabolizing, breaking down the fat in their body, right? They start excreting concentrated urine. All these are physiological adaptations. Now morphological changes, where do you see? You see it in opuntia. Now what is opuntia? Opuntia is nothing but the cactus. Now what is cactus? You can see the leaves are modified into spines. Why? Because they're found where? They're found again in the desert regions. They cannot lose water, right? Leaf has stomata, therefore they lose a lot of water. Therefore what this opuntia has done is it has converted the leaves into spines, right? Now spines, there are no stomata, right? Therefore they do not lose water, right? Therefore what do we see? We see 2 is P, 3 is S, 3 is S, 4 is R, right? QPSR. Do we find the option QPSR, right? Option C. Option C is the right answer. Now this is a very simple question. DNA polymerase of Thermus aquaticus, right? Now this Thermus aquaticus is a bacteria which is found in the hot springs, right? Hot springs. It's found at a very high temperature. It is able to survive at that very high temperature. Therefore what do? We require this enzyme DNA polymerase, right? Where do we use this? We use this in the PCR, right? Polymerase chain reaction, right? To synthesize DNA, right? Therefore it is thermostable. It can tolerate high temperature. The DNA polymerase, Taq polymerase, right? We call it as Taq polymerase. It can tolerate high temperature. Therefore it is thermostable, right? This is the correct answer. Now this is a very interesting question. If a recombinant DNA bearing gene for resistant to ampicillin, right? Ampicillin resistant is transferred to E. coli, right? What are they doing? They're taking a culture, right? This is a culture of E. coli. Now what are they doing? They are trying to insert, right? They're trying to insert this ampicillin resistant gene into the culture. right? Now here, what do you see? Here this culture is full of E. coli. This culture is full of E. coli. Now, once this is done, what happens? You can get two types of E. coli. One E. coli, right, with ampicillin resistant gene, right? Now what has happened? This E. coli has transformed, right, from the normal E. coli to ampicillin resistant E. coli, right? Therefore these are called transformants. What are they called? These are called the transformants. What happened? This was a normal E. coli. They have inserted the ampicillin resistant gene, and these E. coli have taken this ampicillin resistant gene. Now they have transformed. Therefore we call them transformants. Now some E. coli, right, E. coli, they do not take the ampicillin resistant gene. Therefore they are like as it is, right? They are normal E. coli. Now these without ampicillin resistant gene are called non-transformants, right? When they're giving ampicillin resistant, it's not necessary for all the E. coli to take the ampicillin resistant gene. Those which take the ampicillin resistant gene are called the transformants. Those which do not take, right, the ampicillin resistant gene are called the non-transformants. Now if this is clear, now what will happen? These transformants contain ampicillin resistant gene, right? Therefore here if we put ampicillin, they contain resistant gene, right? Ampicillin resistant gene. Therefore these will survive. Why? Because they have a resistant gene. They can resist the ampicillin, toxicity of the ampicillin. Therefore they will survive. While the non-transformants, here they do not have ampicillin resistant gene. They do not have ampicillin resistant gene. Now if these transformants are coming in contact with ampicillin, right, what will they do? They'll die. They'll die because of toxicity, right? They'll die because of toxicity. Now what is the question? If a recombinant DNA bearing gene for resistance to ampicillin is transferred to E. coli cells, host cells become transformed into ampicillin resistant cells, right? This is what they are called, transformants. What happens when these E. coli are grown on a medium containing ampicillin? Now they're adding ampicillin and they're growing these E. coli cells. Now what happens? Non-transformants will grow and transformants will die. See, it's the opposite. Non-transformants do not have E. coli. They're minus ampicillin resistant gene. Therefore they'll die because of the toxicity. Transformants will die is wrong. Non-transformants will die, yes. Transformants also will die. This is also wrong, right? Both non-transformants and transformants will die is wrong. Both non-transformants and transformants will grow, right? This is also wrong. Non-transformants will die, transformants will die, non-transformants will grow, transformants will grow. So what has happened is there is a typo here. So the typo here, instead of die, it is grow, right? Instead of die, it is grow in your paper, right? Now what happens? Non-transformants will die, transformants will grow, right? Because they are resistant to ampicillin. Therefore option B is your right answer. Now look at question number 12, right? Which one of the following is based on the principle of antigen-antibody interaction? Based on the principle of antigen-antibody interaction, right? Now you know PCR. What does PCR do? PCR, if you take a small bit of genetic material, any nucleic acid, what will it do? It will give you many copies, multiple copies, right? Therefore this is not related with this principle. ELISA, enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay. Now what does this do? Right, what happens? We already know that if there is a disease, right, particular antibodies are produced for a particular antigen, right? Therefore what will happen in ELISA? They'll take a beaker, they'll put the antibodies specific to that particular antigen, right? Now what will they do? Now this is, for example, let us say this is antigen for HIV, antibody for HIV, right? Now what happens if the person is infected with HIV? the body, right? In the body, there are particular antigens that are produced. Now, whenever the blood is taken and is sent here, when it comes in contact, right? When this antigen, right, is matching with antibody, right? Then there is a substrate here, right? There is a chemical here which will give color, right? It produces color, simple, right? Therefore, ELISA works on the principle of antigen and antibody interaction. rDNA technology is what? You are bringing recombinant DNA technology, you're bringing a foreign gene or a foreign DNA, right, into the genetic material of a particular organism, generally bacteria, right? rDNA. Gel electrophoresis is separation of the genetic material or nucleotides or even proteins, right, based on the density, right, density and the charge, right? Therefore, ELISA works on the principle of antigen antibody interaction. Clear? Now let us look at question number 13, which is a direct question. Which among the following is used to treat emphysema, right? What is the answer? It is treated by option C, human protein alpha antitrypsin, right? It's a direct question from NCERT. Now question number 14 is a very simple question that you've been learning for, since your school days, right? Homeostasis. Homeo meaning what? Same or constant. Stasis, right? Homeostasis. What is it? Maintaining a constant internal environment in an ever-changing external environment, right? Maintaining constant internal environment in an ever-changing external environment. Maintain a constant internal environment in an ever-changing external environment. Maintain a constant internal environment in an ever-changing external environment. Here, this option, do not maintain a constant internal environment, right? That's option B. Now let us look at maintaining a constant internal environment in an ever-changing external environment would be the right option. But let us look at others also. Do not maintain constant internal environment is wrong. Change their internal body according to their external environment. This is also wrong, right? Change their internal environment when the external environment remain constant is also wrong. Homeostasis is best defined by maintaining the constant internal environment in ever-changing external environment. Now look at question number 16, right? Question number 16. Now if you look at question number 16, bacterial diseases among the following, right? Now which are the following bacterial diseases? Brown rust of wheat is caused by Puccinia, right? Which is nothing but a fungi. Correct? Tobacco mosaic disease is caused by virus, right? Tobacco mosaic virus, right? Black rot of crucifer is caused by Alternaria, right? It's also a fungi. Alternaria. Now, late blight of potato is caused by Xanthomonas. It's caused by Xanthomonas species, which is a bacterial disease. It is a bacterial disease, right? Therefore, option D is the right answer. Now look at question number 17. Now this is somewhat 2 or 3 related from your NCERT, all others are the general knowledge, right? So it's a very simple question. Single celled protein is spirulina, right? And vitamin A is carrot, right? Now if you don't know all others, it's fine. Using this you can find the answer, right? But you can also answer it. Beans is protein, vitamin C is in bitter gourd, right? Do you find this answer? 1 is R, only one option. 1 is R, 2 is S, 3 is P, 4 is Q, right? Option B. If you know one single cell protein, you could have got the answer for all the others, even though if you didn't study it in your NCERT, right? So let us look at next question. Look at this. It's a very simple question. Even a small school going kid can answer this, right? It's a very easy question. A chemical substance which is produced by some microbes, a microbe is producing it, which can kill or retard the growth of other microbes, right? What is it called? Simply anti meaning what? Against. Against. Biotics meaning what? Against life. Correct. Therefore, the answer is option D, antibiotics. Correct? Now this question number 19. Select the correct statement from the following. Methanobacterium is an aerobic, right? Is an aerobic. What are they asking you? They're asking you to select the correct, right? Methanobacterium is an aerobic bacteria found in the rumen of cattle. Now rumen of cattle is correct, right? But this is anaerobic. It's an anaerobic bacteria. Biogas is produced by the activity of, again here they have given it as aerobic, it is anaerobic. So this is wrong, this is also wrong. Biogas is pure methane. It's wrong because biogas is methane plus carbon dioxide plus you get all other gases also, right? Other gases, right? Therefore, this is also wrong. Now, activated sludge in the sediment tanks is rich in aerobic bacteria. This is the right answer, right? This is from biogas production and sewage treatment, right? Now, this is a very nice question, question number 20. Which of these enzymes is required to cleave plasmid, right? So very simple question. If you understand what is plasmid, plasmid is what? It's a single stranded circular, plasmid is single, right? Circular, extra chromosomal, right? Double stranded. It's double stranded, right? DNA. It's a DNA. Now look at this, it's in the form of a circle, right? Ligase will do the function of joining. Therefore, this is not required. They're asking you to cleave. Cleave, who will do cleave? Polymerase is also like polymerization, right? Again, this is also joining, polymerization. Endonuclease, exonuclease are the ones which will cleave. Now, what is the meaning of exonuclease? Exo meaning if this is the DNA, this is exo, right? It will cleave here, right? Endonuclease will cleave here in between. Now, a circular DNA will not have open ends, right? Therefore, exo cannot be the answer, right? Therefore, the answer is endonuclease. It can cleave here, here, anywhere. Endonuclease, right? The answer is endonuclease. It's a very nice question. This from the Stanley Miller, right, experiment, spark chamber experiment, right? A very nice question. Stanley Miller stimulated the conditions of prebiotic earth using spark discharge apparatus, right? Spark discharge apparatus. You remember what all he put into it? He put hydrogen, he put ammonia, he put methane, right? All of this. Then, which organic compounds were first observed by him on analyzing the end product of his experiment? Pigments were no, fats were no, nitrogenous bases were no. He observed amino acids, right? He observed amino acids. Look at 22. Most ape-like ancestral primates were what, right? Now, if you look at the evolution of humans in NCERT, they start with Dryopithecus ramapithecus, right? Dryopithecus ramapithecus, right? Now, the answer has to be among these two, Dryopithecus and Ramapithecus. Then comes Australopithecus, Homo habilis, right? Now here answer has to be among these two because Australopithecus and Neanderthal will come later, right? First are the ape-like ones. Now the answer is among these two, they exhibit similar characters, right? They're walking on four legs like the chimpanzees and the gorillas, right? They have body hair, correct? All of this is same, right? They were found around, how many years ago? They were found around 15 million years ago, right? Now among these two, Dryopithecus is the ape-like one, whereas Ramapithecus was more human-like, right? More human-like, clear? More human-like. Therefore, the answer is Dryopithecus, right? Now, what was speciality about Neanderthal man? Neanderthal man has the largest cranial capacity, which was about 1400 cc, right? Cubic centimeters. Then he also covered his body, right? He also covered his body, right? They also buried the dead, right? Therefore, ceremonies were started by Neanderthals, right? Neanderthals. Look at question number 23, the principle of vaccination, right? You've been studying it from school days, right? Vaccination is based on the principle of which property of immune system. Now, what happens? You why do they do immunization? You are injected with a very mild or dead, right? Dead microbes, right? That is vaccine. Now, what happens when these microbes or the dead particles enter into your body will start producing the antibodies, right? Antibodies. Now, whenever you are infected with this particular organism, right, what happens? Your body has already seen what type of antigens these microbes have. Therefore, they already have the ready-made antibodies, right? They have the memory of it, right? Therefore, vaccination works on the principle of memory, right? It works on the principle of memory. It's a very simple question. Now, this is also another simple question. Genome of HIV replicates in the macrophages with the help of an enzyme called what? Now, you know that HIV is an RNA virus, right? But what will it do? It will synthesize DNA from RNA, right? What was our central dogma said? Central dogma said you have DNA. DNA, from DNA you make RNA. From RNA you make protein. Correct? This was what central dogma said. Now we studied all of this, right? Now what is this telling? Here HIV, what will it do? It is an RNA virus. It will synthesize, right? This process was transcription, right? This is translation. This is translation, correct? Now what is this process? If this is transcription, this is reverse transcription. Right? Therefore, the enzyme has to be reverse transcriptase. Reverse transcriptase, very simple. It uses reverse transcriptase to synthesize, right, DNA from RNA, right? Now, question number 25, right? This was a very smart question. You know, they just interchange this morphine and heroin, right? Now, it has to be heroin, and here it has to be morphine, right? Clear? Here it has to be morphine, but what they have done is they have just interchanged it, right? Now, therefore, this statement is wrong. Now, cannabinoids are also known to have an effect on cardiovascular system. This is nice. This is correct answer. Now, cannabinoids are obtained from the plant cannabis sativa, right? From different parts of plant, different cannabinoids are obtained. They use different names, right? Marijuana, right? There are different names for these, right? Therefore, statement 1 is wrong. Statement 2 is right, right? What is the option? Statement 1 is wrong. Statement 2 is right. Option C would hold good, right? Option C would hold good. Now, mule is a result of what? What is mule? This is again out of syllabus. They have deleted this portion from your current NCERT, right? Mule is what? It's a progeny of a male donkey and a female horse, right? They're two different species, right? Therefore, it's a interspecific, meaning what? Inter meaning between, specific meaning species, right? It is interspecific hybridization. Mule is a product of interspecific hybridization. Clear? Now look at question number 27. It's a direct question that was asked from reproductive health, right? The government of India legalized MTP, medical termination of pregnancy, with some restrictions, with some regulations in the year 1971, right? In the year 1971. Now question number 28. The process in which a small part, right, of the vas difference, right, the small part of vas difference is removed and tied through a small incision is called what, right? Very simple question. Vas, right? You're cutting and doing something with the anatomy, right? Tomy meaning cutting, right? Therefore, it has to be vasectomy, right? Vasectomy, right? Anatomy, vasectomy, right? Therefore, it has to be vasectomy. 29. A test cross in pea plant is what, right? A cross between F2 tall plant and a recessive parent, right? Test cross, we know it is always with recessive parent. But what is this? This is of the unknown phenotype, right? Or the unknown genotype, right? You know the phenotype, but you don't know the genotype, unknown genotype, right? Now, for example, here look at the option A. Here a cross between F2 tall plant. Now, tall can be TT right? Or it can also be T and T. Now this we don't know. This is unknown versus a recessive parent which we know. Look at option B, a cross between F2 dwarf. Dwarf we know it is small t. This is for sure with a recessive parent. Therefore this is not a test cross, right? Now a cross between F2 tall plant, right? Okay, tall is again we don't know. Therefore it can be this or it can be this with a dominant parent. Now dominant parent is this and this. When it has to be recessive only, then it's called a test cross. Therefore this is also no, this is also no. A cross between two F1 plants. Now F1 plants and F1, one has to be recessive parent. Therefore option A holds good, right? Because it's between the unknown genotype and the recessive parent is called a test cross. Question number 29. Clear? Now look at question number 30, right? The genotype ratio, the genotypic ratio of incomplete dominance, right? Incomplete dominance is what? Very simple. If you know the answer, it's well and good, right? It is what? It is 1:2:1. Now incomplete dominance, NCERT, we study the snapdragon flower, right? Flower color of snapdragon, right? Now how do we start it? We know that there is a red color parent, there is a white parent, right? Now here both are red, here it's small r and small r. Now they produce gametes r, here it's small r. Now when these two meet in the F1, what do you get? You get r and this is pink, right? Incomplete dominance. Incomplete dominance meaning what? This is not completely dominant. If it is completely dominant, it has to be red, but here it is incompletely dominant, therefore you're getting pink. Now what will happen? You cross F1 into F1, what will you get? You get r into small r capital R. Now here you can see what? Here you write 1 capital R small r, another parent is here capital R small r. Here you can write one is male, another one is female. This also you can write one is male, other one is female, right? Now what do? You get capital R. What would this be? It would be red, capital R small r. You get pink, capital R small r. It is again pink, small r and small r. This is white, right? The speciality of incomplete dominance is you get what? You get both genotypic and phenotypic ratio as same, right? The ratio of phenotypic and genotypic is same. Now what do you get? You get capital R is to capital R small r is to small r. What will you see? You see 1, right? 1 is to, you see pink 2 is to white is 1, right? If you write the phenotype red, pink, white, even then you get 1:2:1. Answer is 1:2:1. Option B, right? I hope you got this clear. Now look at next question. Find the incorrect statement among the following. Find the incorrect statement among the following. In a sex-linked recessive trait, the genes are transmitted from unaffected carrier female to some of the male progeny, right? This is correct. Yes, from the unaffected female carrier, it is being transferred to some of the progeny. They are asking you the incorrect. Accumulation of pyruvic acid in brain results in mental retardation. This is also correct. This is also correct. Individuals affected by Down syndrome will have congenital heart disease, right, or heart defect, right, and more intelligent, right? Now here, this is, they are less intelligent, therefore this is incorrect, and this is the right option. Turner syndrome is caused due to the absence of one of the X chromosome. Yes, right. How do you remember? It is DK Turner, right? Here DK is, he's got plus 21 plus X minus X, correct? Down syndrome is caused due to trisomy, right? An additional chromosome at chromosome number 21. Klinefelter is due to the additional X chromosome, whereas Turner syndrome is due to a lack of X chromosome. One X chromosome has been deleted, right? Therefore, this is also right answer. Therefore, the option C is the correct answer for question number 31. This is a very interesting question. In a dihybrid cross between true breeding yellow seed and a true breeding wrinkle seed, right, green seed plant, the ratio of segregation of round and wrinkle seed traits in F2 is, right? It's a very big question, right? If you know the dihybrid cross, right, from NCERT, right, what do you see? You see there are 16 right, you can see 16 columns in the checkerboard, right, 16 boxes in the checkerboard, and what will you see? You see, what are they asking? They're asking round, right? Now round, you get 12. There are round 12, right? 12 are round. Wrinkled, 4 are wrinkled. 4 are wrinkled. Now if you look at this question, immediately what would you do? You guys would do 9:1, right? But that is wrong. Please look at this. It is 12:4, right? Now you do this, 4 ones are, 4 threes are. Therefore answer is 3:1, right? So this question is, answer is 3:1. Now if you want, we can solve this, right? If you want, you can solve. Round yellow is capital R, capital R, capital Y, capital Y. Wrinkled green, right? Therefore small r, small r, small y, right? If you want, you can solve this question, right? If we take r, capital Y, small r, small y, right? What will be the gametes that they produce? You get capital Y, capital R. Here you get small r, small y, right? Now when they two fuse together, you get r, y and y. Now this is round yellow, right? But when they produce, right, when they produce gametes, right, now we are taking, this is F1, now you take F1 into F1, now you take R, Y crossed with R, Y and Y. What are the types of gametes they produce? Each one will produce 4 types of gametes: R, Y, R, Y, R, Y, R, Y. Right here also you will get R, Y, small r, capital Y, small r, small y. Now if you do this, you do the entire checkerboard, right, you will get, right, 12 are round, 4 are wrinkled. Simply pick the NCERT and look at the table, right, the checkerboard of dihybrid cross, and you will be able to see, right, this, they are round are 12 and 4 are wrinkled. Now look at this question. This is a simple question from Griffith's experiment from molecular biology, right? Very interesting question. But before we go, you need to understand there are R strain and S strain, right, of bacteria, right? Pneumonia causing bacteria. Now S strain is virulent. This will cause disease. S strain is virulent and it will cause the disease, right? Because it has a smooth virulent coat. Clear? Now R strain is non-virulent and it does not cause any disease. This is the basics. Now R strain is injected into the mice will die. Wrong, because R strain is non-virulent, it will not cause the disease. S strain heat killed. Now you have killed the virulence, right? Heat killed is injected into the mice and mice dies. This is also wrong, because once it is heat killed, this effect of disease is gone, right? Now R strain injected into mice alive. This is correct. Now which of the following is correct? Only R is correct. Option B. Option B is the right answer. Clear? Now look at question number 34. In tRNA, the region that binds to mRNA is what? Right? It's a very simple question. You know tRNA is a cloverleaf structure like this. Here there is a variable arm. Right? This is the 5' end. This is the 3' end. You have the CCA end. This is where amino acid will bind, amino acid binding region. You have the T loop, right? This is the D loop. Now this is where, right? This is mRNA. What does mRNA contain? mRNA contains codon, right? Here this is the anticodon loop. Anticodon loop, right? Therefore, where does the tRNA, in the tRNA, the region that binds to mRNA is the anticodon loop, option A, right? Now look at question number 35. The mRNA has untranslated regions, right? If you look at the structure of mRNA, right, this is mRNA, it has a 3' end, it has a 5' end, correct? Now what you see is here, right, here, after the 3' and a 5' end, you see UTRs, right, untranslated regions, right? Now why are they required? They are required for efficient translation, right? Efficient translation, clear? They're required for efficient translation. So therefore, where do you see UTR? You see it on either side of the 3' and the 5', right? On the after 3' and 5', right? So you can see this option C at both 3' and 5' and beyond terminator codon and before the AUG respectively, correct? Before the terminator codon and beyond the terminator codon and before the AUG respectively, right? Therefore option C is the right answer. Now look at question number 36, a very simple question, right? If you simply know that there is base pairing, right, you're good to go, right? In the structural gene, the template gene, right, DNA strand nucleotide has nucleotide sequence 3' to 5'. Find the correct complementary nucleotide sequence on the coding strand. Now if you look at the transcriptional unit, right, if you look at the structure of transcriptional unit, it is 3' to 5', right? Here it is 5' to 3'. Now here there is promoter region, here there is terminator region. Now promoter is like this, correct? Now this is the template, right? Now this is what they have given, right? ATGC. This is coding, right? Here this names are given, it's not like the proper ones, right? They're given as a misnomers, correct? Now this is coding. Why? Because this is the template. Now, whatever complementary it is similar to this, and that is the mRNA, correct? Therefore what happens if you just forget everything and just see this is 3', 5', 3' and 5'. Therefore what should the answer be? Answer should be 5' to 3'. There are two options. Here it is A will pair with AT. Among these two, who is there? It is the option C. Just with one you can see T will pair with A, G will pair with C. Therefore option C is the right answer, right? Very simple. Now you can look at this question. This was asked from evolution, embryological evidences of evolution, right? Now statement 1, all vertebrates develop a row of vestigial gill slits during the embryonic stages, which is correct, including humans. You see it in all vertebrates, but these vestigial gills are functional only in case of fishes, right? All others, they are vestigial, right? This is right. Embryos always pass through the adult stage of other animals. Now this is wrong, right? If an embryo passes through the adult stage of other animal, now it, see, for example, let's say an embryo of a rat is passing through the adult stage of a human, it will become a human, right? It's not a rat anymore, right? Therefore it is wrong. NCERT says embryos will never, this always is not there, it is never. Embryos never pass through the adult stage of other animals. Therefore this is a wrong statement. Now option A, statement 1 is correct, statement 2 is incorrect, right? Clear? Very simple. Ciliated epithelial cells, where are they found? It's a direct question. Ciliated epithelial cells are found in the bronchioles, right? They're found in the bronchioles. Look at question number 39. Which one of the following statement is correct with reference to the vacuole? Now if you study, this question is from the cell, the unit of life, right? Vacuole, you see it in plant cells, mostly in plant cells. You also see it in euglena, right? These type of things. Amoeba, you also see it in amoeba, right? Now vacuole is a single membrane, it's a single membrane bound organelle, right? It's a single membrane bound organelle. What does it contain? It stores a lot of water. That's the reason you see the nucleus in the plant cell is moving aside, right? It's pushed to one side because the vacuole contains a lot of water, right? Other excretory based products, right? Excretory based products from the plant. Therefore it's a membrane bound and contains storage proteins and lipids. Is wrong. It's a membrane bound. What is a membrane called? The membrane is called the tonoplast, right? And contains water and excretory substances. right? This looks right. It lacks membrane. No, it has a membrane, right? It lacks membrane. This is wrong. Option B is the right answer, correct? Now look at 40. Exoskeleton of arthropods is made up of a unique complex polysaccharide, right? Which is a polysaccharide? It is chitin, right? It is chitin, correct? It's chitin. The enzyme recombinase is required at which stage of meiosis? Now, if you look at prophase 1 of meiosis, right? If you look at prophase 1 of meiosis, you know prophase 1 is divided into leptotene, zygotene, pachytene, diplotene, and diakinase, correct? Now what happens in leptotene? Chromosomes become visible. Correct? Now in zygotene, what happens? The homologous chromosomes will start pairing together. Right? Then they come in contact with each other. What do we call it? We call it synapses, right? Pairing together, synapses, and it takes place with the help of a protein called the synaptonemal complex. Correct? Now in pachytene, what happens? Pachytene, recombination nodule is formed. At the point of contact, which contains an enzyme called recombinase, which brings about crossing over, right? In diplotene, what happens once the crossing over is done, right? Chiasmata, an X-shaped structure is formed. Then in diakinases, terminalization of chiasmata occurs and the two homologous chromosomes will separate, right? This is in brief whatever is happening in meiosis 1, right? Prophase 1 of meiosis 1. The enzyme recombinase comes in contact where? It comes in pachytene, because recombination nodule is formed and crossing over occurs here, right? So it's in pachytene. Now which one of the following exhibits haplodiplontic life cycle? Which one of the following will exhibit haplodiplontic life cycle, right, is the question, correct? So what do you see? Fucus will show diplontic life cycle, right? Chlamydomonas will show haplontic life cycle. This is also from the deleted portion. Gileadium will show haplontic life cycle. Your ectocarpus will show haplodiplontic life cycle, right? Therefore, the answer is ectocarpus. Now, this is from the deleted portion. This is not taught to you, right? It was deleted from your syllabus. What is this diplontic, haplontic life cycles? You know, plants show two life forms, right? They exist in sporophytic form and they exist in the gametophytic form, right? In case of haplontic life cycles, right, here the gametophytic life form is dominant, right? And sporophytic form is seen only in the form of a unicellular zygote. Only zygote is seen in the sporophytic form. Whereas in case of diplontic life cycle, right, here what happens, right? Here also the forms might vary, a sporophyte and gametophyte, right? But here the dominant phase is sporophytic phase will be dominant, right? You can see them as a dominant phase. Few sporophytic is majorly seen, right? Here ectocarpus, they show equal, right? Equal dominance of both haplontic and diplontic life cycles. Clear? Now here this question is from the animal kingdom. Animals, identify the phylum which shows the following characteristics. Animals are exclusively marine, radially symmetrical, and diploblastic, right? Body bears 8 external rows of ciliated comb plates, right, which help in locomotion. Digestion is both external cellular and intracellular. Reproduction is only by sexual mode. Where do you see this? You see it in Ctenophora, right? You see this in the Ctenophora, phylum Ctenophora. Now look at question number 44, another simple question. A flower which has both stamens and carpels is called what? It's called bisexual flower, correct? 45, a typical lub-dub sound here heard during heartbeat are produced due to what, right? Here what happens, you know, heart, there is right atrium, right, and right ventricle. You have left atrium and left ventricle, correct? Now between these two, what do you see? You see tricuspid valve, right? Between these two you see the bicuspid valve, right? Bicuspid valve. Now here from the ventricle to, right here, what do you see? You see the semilunar valve, correct? Semilunar valve. Now when do you see this lub dub sound happening? The closure of bicuspid and tricuspid, right? These two together will form the lub sound. Now semilunar valve will form the dub sound, right? That's the second sound. Clear? Therefore the option is C. Look at 46. The functional unit of contraction is what, right? Center of Z zone, a portion of microfibril between two successive Z lines, the portion of microfibril between two successive M lines, the center of Z zone, H zone, and the center of I band, right? Now what do you see? You see that if you remember the mechanism of muscle contraction, these are the Z line, right? Right, what do you see? You see sarcomeres like this, right? Then there is H. Correct? Now this is H zone. Here this entire thing is A. Correct? Now when these contract, right? When these contract, right? What happens? There is contraction in the muscle, right? Therefore the answer is portion of microfibril between two successive Z lines, right? Is causing the contraction, right? Causing the contraction. Now look at 47. They've given the parts of brain and they've asked you to identify the functions of the brain. Now if you look at this, right, you know medulla oblongata is helping in respiration, hypothalamus helps in body temperature, regulation of body temperature, cerebral cortex helps in motor function, limbic system helps in olfaction, right? Very simple question. Therefore you see 1 is R, do we have it? 1 R, 2 is P, 3 is S, and 4 is Q, right? Option D is the right answer, correct? Look at question number 48. Hydra reproduces asexually by producing what, right? You know Hydra, right? It's a coelenterata. Now if you look at the structure of Hydra, right, somewhat like this, right, like this, what does it do? It will produce small buds, right? Buds. It will produce budding, right? Budding is a process, right? Therefore it produces buds, right? That's a simple answer. 49. If the male gamete and the female gametes are morphologically distinct, right? Now this is also from your deleted chapter, right? Second year, first chapter is deleted reproduction in organisms. Now here, but it can be considered because this are discussed, isogametes, heterogametes, homogametes are discussed in algae, right? Therefore I don't know if they're going to delete this question, right? But this is considered in algae, right? Therefore what are homogametes? Homogametes are both male and female are alike. What are heterogametes? Male, female, and male, right? The difference in look, right? Morphologically different. They're called heterogametes, right? Hermaphrodites, you know, a single organism having both male and the female reproductive system. Sexual dimorphism is by looking at morphology, you can distinguish whether it's a male or a female. Therefore, answer is B, right? Answer has to be B. Now water potential of pure water is what? Now this is also again from a deleted chapter, mineral nutrition, right? Now water potential of pure water, right? Water potential of pure water is 0, right? It's 0. Now why is it 0? Because see, one person can sell pure, what is water potential? Water potential is basically purity. Purity of water. Now what do? One person says purity of water is 100%. Another person said purity of water is like 1000. My scale of measuring purity of water is 1000. Another person is telling my scale of measuring purity of water is 100. Another person will tell my scale of measuring purity of water is 10. Now to avoid this problem, what did we do? They said purity of water 0 is the purest form, right? Is the purest form of water. Water potential is 0, then the water is purest form, purity of water, right? Therefore water potential of water is 0, right? Question number 51. Match the pigments given in list 1 with their color in the chromatogram given in list 2, right? Now what do? You know chlorophyll B is, right, yellow green, chlorophyll A is blue green, xanthophylls are yellow, carotenoids are yellow orange, right? Now look at option B. Option D, we checked it, right? 1T, 2P, 3R and 4S. This is wrong, right? Here there is a mismatch. Therefore, we are looking at option B. 1T, 2P, 3S, 4R, right? Now this is the right answer. Clear? Right answer. Look at next question. Which one of the intermediate compound that links the end product of glycolysis with TCA cycle, right? Now, if you know glycolysis is a process where breakdown of glucose occurs, right? Breakdown of glucose will occur and it will result in product. What is the product? You get two pyruvic acids, right? Now, glycolysis will result in two molecules of pyruvic acid. Now, this pyruvic acid by link reaction, what reaction? By link reaction with the enzyme, help of enzyme pyruvic acid dehydrogenase, right? With the help of an enzyme pyruvic acid dehydrogenase, it will get converted into two molecules of acetyl CoA. Acetyl CoA, this is the intermediate. Now this intermediate will enter the TCA cycle, right? Therefore, what is the intermediate? Intermediate is acetyl CoA. Acetyl CoA, correct? Very simple question. Oxen will show apical dominance, right? It produces apical dominance. Gibberellin will produce what? Gibberellin will produce bolting, right? Elongation, shoot elongation before flowering is a character, important character of gibberellic acids or gibberellins. Now 54, the term uremia, right? Uremia is what? Accumulation of urea in the blood. Do we have it in the options? The first option itself is uremia, right? Option A. 55, which one of the hormone is not secreted by the human placenta, right? If you know, in human reproduction, you study the human placenta and you study the endocrine function of human placenta. The endocrine function of human placenta is to secrete, it will secrete HCG, right? Human chorionic gonadotropin. It will secrete HPL, human placental lactogen. It will also secrete some small amount of estrogen and progesterone, right? Now, which of the following is not secreted, right? Progesterone, HCG, estrogen are secreted by placenta. LH is secreted by the pituitary, right? Therefore, LH is secreted by pituitary and not by human placenta. Now, in human females, the endometrium of the uterus consists of what, right? It's a very simple question. You know, uterus is made up of three layers, right? First one, outer, right? It's made up of outer perimetrium, right? Outer perimetrium. This is thin membranous, right? This is thin. This is thin membranous. You have the second layer, which is the middle myometrium. This is made up of thick, smooth muscles, right? Then you have the third one, it is inner endometrium. This is glandular. It's a glandular layer, right? Now, what is the function of middle layer? It will undergo rigorous contraction during child labor, right? Or the birth, parturition, right? Or delivery. Now, inner endometrium will undergo cyclic changes during the menstrual cycle, right? Therefore, the answer is glandular layer. Glandular layer. Very nice question, right? If two primary spermatocytes and two primary oocytes undergo meiosis simultaneously, what will be the ratio of spermatozoa and ova produced at the end of gametogenesis, right? Very simple question. We know one primary spermatocyte will produce 4 sperms at end of spermatogenesis. We know one primary oocyte will produce 1 ova at end of oogenesis, correct? Now, what are they telling? They are taking multiplying it by 2, right? Now, what will you get? You get 8 sperms and 2 ova our ratio is 8:2, 1s are 4s are. Now the ratio still remains 4:1, right? 4:1. Now look at question number 58, the role of filiform apparatus in synergids, right? Where do you see? You see synergids in the embryo, right? Embryo sac, right? You see it in the embryo sac. Now if you look at synergids, this is the embryo sac. You see on the chalazal end there are 3 antipodals. In the micropylar end, you see 2 synergids behind which there is egg, right? This is egg. Now these 2 are the synergids, right? Synergids. Now here you see filiform apparatus, right? The cellular thickenings. These are filiform apparatus. Right? Now if you see, this is at the micropylar end, right? This is the micropylar end, correct? This is a micropylar end. Now why are these 2 together we know are called egg apparatus? Right? Now why are these egg apparatus present at the micropylar region? Because the pollen tube has to enter through the micropyle. And what will these filiform apparatus do? They'll guide the pollen tube into the embryo. They guide the entry of pollen tube into the embryo sac, right? Embryo sac. Now transfer of pollen grains from anther to the stigma of another flower of a same plant, right, is called what? Another flower of the same plant. Now if you remember, this is a flower, this is another flower, right? This is another flower of a different plant, right? Now if the pollen is transferred within the same flower, we call this as autogamy. Autogamy. If the pollen is transferred to a different flower but of a same plant, right, then we call it geitonogamy, right? If the pollen is transferred from one flower of one plant to a flower of another plant, then we call the term xenogamy, right? Therefore the answer here is transfer of pollen grains from anther to the stigma of another flower of a same plant, right? They're talking about this geitonogamy. Option D is the answer. Now question number 60, we reached the last question. It's a very nice question and a simple question from sexual reproduction in flowering plants, right? Polyembryony is what? Poly meaning many, embryo, more presence of more than one embryo in seed is called polyembryony. Where do you see this? This is very common in citrus and mango, right? Perisperm is the remnant, right, or the remaining nucellus in the mature seed, right? We call perisperm. Where do you see it? Beet and black pepper. False fruit is a fruit that is developing from any other part of a flower except the ovary. Here thalamus develops into fruit in case of apple. Parthenocarpy is the production of fruit, formation of fruit without fertilization. Parthenocarpy, where do you see it? You see it in banana, right? Do we have the option? 1 is R, 2 is P, 3 is S, 4 is Q, right? R, P, S, Q, option A. Okay, so this is the answer key discussion in detail for your CET paper 2024 biology, right? I hope you understood this and you got all your answers right. Okay, before you sign off, please like, share, and for more content please subscribe to our channel Diksha Karnataka. Okay, thank you students, have a great time.